Welcome everybody to the March 24th meeting of the working group. I appreciate you all for coming. We're very grateful to our speaker for being willing to speak today to us from the USDA. As always, I need to remind you that this meeting is recorded and live streamed, so please keep that in mind. As always, also, Reinhard and myself are available for criticism, comments, suggestions. Jim Sluka and Lorenzo are also always here to help try to make things work better for you. We have our usual communication channels. Please do help us let people know about these meetings. Especially, we would appreciate your help getting word out about our large and growing archive of videos of wonderful talks. We have more than 100 seminars available on our YouTube channel, covering all sorts of interesting areas. Please help make sure that people know they're there. We do have our Twitter feed, MSM Viral. We would very much appreciate your help retweeting our announcements and getting the word out. For announcements today, I have two. One, we have our usual Digital Twin subgroup tomorrow at 11 a.m. Eastern Daylight Time. If people need the link to that and are not, don't have it already, please let Reinhard or me know. We're happy to send that out to you. That's open to anybody who wants to participate. I also was posting something that came across my desk today, which is that the Human Frontier Science Program has a solicitation due May 12th for postdoctoral fellowships. And these are international fellowships. The rule is that you have to change country. So if you did your PhD in the United States, you could go to Europe. If you did your PhD in Europe, you could come to the United States. Traditionally, the US has not been a destination for these. They seem to have opened up the nationalities quite a bit. I would think many people within this community might potentially be able to send students to other groups through that program, or vice versa, try to recruit students through that program. And so I encourage you to take a look at that. It's a great opportunity. It's quite competitive, but historically, computational modeling of biological systems is something they do and support. So I encourage people to consider that. Are there any other announcements before we go on to our talk? No.
This is Reed. I could just drop a quick funding announcement in the chat from CDC. Maybe there are people in the CDC that know more than me about it, but it's for genomic centers of excellence, not directly related to modeling, but there might be some modeling components. It's looks like a lot of money, so I'll drop it in there and you guys can check it out.
Great, thank you, Reed. We appreciate that. Heads ups on RFAs from NIH or elsewhere are always appreciated. This is a hungry crowd. Okay, coming up, we have March 31st. We're going to try a discussion. We hope you'll show up. Reinhard and I are going to talk about the future of the working group and try to think about what we can do to be more effective and to work better. April 7th, we have 2 speakers lined up. We are still looking for speakers starting from April 14th. As always, people have lots of great ideas, but when I ask them to suggest names, they tend to be quiet. The cost to you is very low of suggesting you see a paper that you love and you'd like to hear from the speaker, from the authors, please just send us the names. It could be experimentalists, clinicians, drug developers, modelers, people from the USDA, anyone who has anything connected to the idea of understanding pathology and immune response in response to infection is fair game. We could have another journalist. That would be interesting. We could talk about social consequences. Anything that you find interesting. And as always, I will remind you, please suggest yourselves as speakers, your graduate students and postdocs. this is a great opportunity for people to make connections. Please try to remember to mute your mics. We only have one talk today, so we have questions right after our talk. And our seminar today is from Susan Schreiner from the USDA in Fort Collins. And we are going to be hearing about influenza infection dynamics in wild birds. And I will turn it over to her.
Today, well, I'm going to give a very slight background. I work at the National Wildlife Research Center. We're in the Animal Plant Health Inspection Service and specifically in wildlife services. And what we do there is a combination of experimental work, modeling, and we do a lot of surveillance work. And so we do surveillance for pathogens of interest in wildlife. The last few years, a lot of that has looked like looking for SARS-CoV-2 in different wildlife hosts. But my project specifically focuses on emerging diseases. And what that's meant for me for most of my career has been a focus on avian influenza viruses. And we're interested in understanding the natural ecology of influenza viruses in the wildlife hosts so that we can understand potential impacts to poultry, since I am in USDA. And so today I'm going to focus on the more experimental side of the work that I do. We are very lucky in Fort Collins because we have really excellent facilities for doing experimental work with pathogens, and we can work with wildlife hosts that have requirements that are a little bit different than working with lab animals. And so the main experiment I'm going to talk about today is the influence of exposure dose on infection kinetics in mallards, and then a couple of follow-up experiments we did after this. And our motivation for this work has been, give me a second, there we go. Our motivation for a lot of this work is so that we can parameterize epidemiological models and do some scenario analyses or agent-based models so we can identify control points to try to prevent the spread, to be able to predict potential spread to poultry and to protect poultry and prevent spread. And so for influenza virus, everyone's familiar with influenza. It's a threat to poultry industry, but it's also a public health risk, and in some cases, a threat to wildlife. I don't know if you're aware, but we are in the middle of a very intense incursion of high-path avian influenza in the United States right now. It came in wild birds, and now it's spreading pretty fast throughout the nation. I think we have 27 states, commercial operations, as well as hundreds of detections in wild birds. And so this is the kind of thing that we're trying to, the more we know about the natural ecology of these viruses in wildlife in North America, the better we can do when these things happen. And so I have done a lot of work studying mallards, and there's some good reasons why I focus on mallards. It's a very common influenza reservoir host in the United States. In these two maps on the left, we see what is called breeding bird survey data. So those are data collected on the numbers of mallards in the United States during the breeding season. And then on the right, we have the same kind of study done in the winter. And you can see that it's a very common species in North America. But one thing that's important to look at these two maps is to see the changeover between these two maps. And because mallards are a migrating species, what that means is that we have these huge migrations as they come to overwinter in the United States. And so when they overwinter and then migrate in, they're bringing all their viruses with them. And so we get these huge mixing of viruses in the fall. And so we really need to understand that through surveillance, we can help inform that with our experiments. And so influenza in the United States, we see peak in wild birds in the fall, and then it kind of tapers off. And then we see it's very cyclical in these populations. And not only are mallards very important reservoir hosts in the United States, but they're globally distributed, especially in temperate areas. And so they play an important role in the natural ecology globally. And so the question here is, we want to be able to compare, and when I try to model influenza dynamics, is our basic infection curve, and I look, does that infection curve change over time, change in response to different doses of exposure doses. And so we want to break that down into the different subunits of the infection curve. And so here, I'm assuming you're familiar about, the animal is exposed, and then we have the eclipse period, which is the period before the animal starts excreting the virus. There's all kinds of things that have to happen. The virus has to attach to the cell membrane. It has to get into the cell, then it has to get into the nucleus. Once it's in the nucleus, it kind of has to harness the machinery of the host to start replicating the virus. And then there's a whole set of things that have to do for that virus to get excreted. And so we have, all kinds of shapes of these different infection curves. So we have that eclipse phase, and then the virus starts replicating in the host, and that can grow very fast. And then you can get this kind of peak period. Sometimes it's narrow and tight, sometimes it's broad, spread out depending on the pathogen, but even within influenza, it can be very different across strains and hosts. And then we get this kind of display period. I think that was my next one. Okay, so enough background. Moving on to my methods. There's a pretty straightforward experiment. We experimentally inoculated mallards. We used an H6N2 virus, which came from our surveillance, so an endemic virus from wild birds. And then we exposed mallards to 3 different doses, 10 to the 3, 10 to the 4, and 10 to the 5. And I just want to point out here, this is one of the things that we really don't always understand, is we don't know what's happening naturally, what are natural exposure doses. And so we're always kind of taking a guess at what that might look like. I've studied, influenza and mallards for a long time, and I really think it's probably very broad, because influenza is a gastrointestinal disease for birds, for most birds. For Galaphones, it's a little bit more respiratory like humans, but it's a gastrointestinal disease. And so one of the major things that they do is they excrete the virus in their feces. And so if they're in the water and they excrete, and there's another mallard right behind it, they could be exposed to a very high dose. On the other hand, if the feces has a while to dilute in the water, they could be exposed to a low dose. So we don't really know what those natural exposure doses are. So we're kind of guessing when we do these experiments. We used reproductively mature mallards. And then what we did was we had 10 groups, and for each group, and these are in separate pens, we had 1 inoculated duck, and then we had 3 naive contacts with that inoculated ducks. So for the inoculated ducks, our sample size was 10, and for the contact, our sample sizes were 30 across those 10 different inoculated ducks. And so there were 4 ducks in each pen, and they were separated from each other. And then we collected swabs, oral, coecal, and fecal swabs from the birds across 10 days. And then we sampled those, we tested those with PCR. And so I want to bring your attention to the graph here, just to show what our sampling scheme looks like. We wanted to get very fine scale information on what these infection dynamics look like. And there was a couple of reasons. One was because we wanted to set up a transmission experiment. And so we wanted to know what was the minimum time for when did that first excretion happen, because we wanted to be able to control to make sure that ducks that were for the transmission experiment were getting it from the main individual and not from each other. And so that was one thing. But we also noticed that in this kind of standard way that people do these experimental inoculations, sometimes they only look every day, like every 24 hours, or they evaluate sometimes only every other day. But it seemed to us like there was a good chance that those peaks were occurring prior to day 1. And so what we did was we inoculated the animals, and then we tested them every 2 hours for the first 24 hours. And then the second day, we sampled them every 8 hours. Every 4 hours. And then on day 2 and 3, we sampled it every 8 hours. And then just once a day after that. From prior experiment, we knew that the main infection dynamics happened in those first few days. And so here are the main results of that study. Just to get you oriented, we have the results from the oral, the coecal, and the fecal swabs going across. And you'll see that on the top. The x-axis is the hours post-inoculation, and the y-axis is the quantity of virus that's being excreted, at least viral RNA from PCR. And so the main thing we see is that, we see the general infection curve across, but hopefully we're just seeing the means here, but you see that there's a lot of heterogeneity in what these curves look like. The 3 different curves for the 3 different doses are in the 3 colors that you see, with the 10 to the 3, the low dose, being in gray, and then the 10 to the 4 and 5 in orange and blue. And so if you look across these 3 graphs, you'll see that the 10 to the 4 and 10 to the 5 dynamics are very similar across the infection, whereas the 10 to the 3, the dynamics look quite a bit different. And in general, for the 10 to the 3, what we see was it took a lot longer for that virus to really take off before the animals were excreting. And then once they excreted, it was a little bit lower. The one thing that we found is for the fecal samples in particular, we probably ended this experiment just a little bit early at that 10-day mark. And they might have excreted a little bit more because they weren't, all the animals weren't down to 0 on 10 days. So that slower group. And so, these graphs are all for the animals that we directly inoculated. But then remember, we had 30 birds where they were being exposed to these individuals. And so what was really interesting to us is that we saw very similar dynamics across those 3 exposure doses, even in the contacts. And so that was pretty interesting, where if you look at that first one, the 10 to the 3, we have much slower dynamics. And so in these graphs, same axes, but the inoculated birds are in gray. And so that's what you just saw. That's just the mean of what you saw on the other page. And then the orange is the transmitted. And so they look very similar. So that was interesting. So what it tells us is that those higher exposures, they're excreting fast and quickly. And so the birds are being exposed at higher doses. But when the birds took longer to start excreting and it didn't have such a steep rise in the excreted virus, the birds were likely exposed to a lower dose. And so we saw the infection dynamics in the transmitted birds were very similar to the inoculated birds. And so that was pretty interesting. This chart just shows you kind of like honing in, because that fine scale data kind of gets lost in those overall results I just showed. And what I want to kind of emphasize, this is just showing the first couple of days of the experiment. As you see, there's a lot of variability, but what I really want to emphasize is just how quickly the infection occurs in these animals. For what we're seeing here are the results for the animals at 10 to the 5. And on the top 2, we see the fecal shedding for inoculated and transmitted birds. And in the bottom, we see shedding in the oral cavity for the inoculated and transmitted bird. So at this 10 to the 5 level, the fecal shedding happens so fast that they were, the first detection was at 10 hours post-exposure. That was, pretty fast. And the animals were already shedding at 10 to the 6 in less than 24 hours. And so so they start shedding, like we saw those first, very, early indicator of the replication at 10 hours. And then by 20, it's really starting to get kind of our exponential growth in those infection dynamics. Another thing to note is, as I mentioned, this is a gastrointestinal disease. We see the most excretion in feces. And if you look at the oral, they actually started, it's hard to know because we inoculate them orally, that's what we think happens with ducks, is they probably drink in contaminated water, and that's how they become exposed. And so they were shedding a little bit faster, but the virus hits those tissues faster, but they never reached the same peak. They peak around 3.5, 10 to the 3.5 to 4, compared to the feces where they're excreting at 10 to the 6. And sometimes higher, depending on the virus we use and other factors. And so anyway, these dynamics are very fast. And so I'm going to kind of quickly go through some of those things that we looked at here. This chart is showing us the results from that eclipse period. And this is all going to just mirror what we see from that bullet curve. So for the eclipse period, it was much longer period. So we have hours post-inoculation on the y-axis and then the dose on the x-axis. And in all of these, the colors are just the 3 different swab types, the oral, cloacal, and fecal. And so we could see the eclipse period was much more spread out for the 10 to the 3 group, but we see pretty similar dynamics in 10 to the 4 and 10 to the 5. For the peak day and the peak load, same thing. The peak day occurred later for the 10 to the 3 mallards, and the peak load was lower for the 10 to the 3. But for the 10 to the 4 and 10 to the 5, those doses were pretty similar. And then for the total load, so not just the amount of virus excreted on the peak day, but across the entire infection, we do see that 10 to the 4 and 10 to the 5 are very similar. But in this case, the 10 to the 3, even though those dynamics were slower, they did excrete across the entire infection period almost the same amount of virus. It's a little bit lower, but it's a little bit confounded because those animals weren't done excreting when I ended the experiment at 10 days. But obviously I didn't have that information when I designed the experiment. And then as far as infection duration, like how long on average did we see the birds excreting virus, or at least excreting viral RNA? Again, 10 to the 4 and 10 to the 5, very similar. And that period was, it's actually pretty similar, but just a little bit lower. But again, it might've been stretched out by a day had I continued the experiment. And so it's a little confounded there by my experimental design. So in summary, for this part of the experiment, we did see that dose mediated the infection characteristics, but probably there was a saturating effect where the 10 to the 4 and 10 to the 5 were very similar. And then something that we thought was very interesting was that the transmission infections compared to the direct inoculation recapitulated the exposure dose. So those infection curves for the contacts was very similar to the directly inoculated controlled dose. We had very rapid infection dynamics with the birds shedding within 10 hours and transmission occurring within 24 hours. And the one thing I really want to emphasize for when people do modeling is just that we saw such significant individual heterogeneity, which can be very important for doing like agent-based modeling or anything like that, that we have to really account for that heterogeneity. So now I'm going to go on, and this is kind of like a separate talk, but it's really just furthering that experiment. So when we did those experiment with the infection dose, we were kind of guessing where to start. And we started at 10 to the 4, and then we moved to 10 to the 5. And then after we did 10 to the 5, we thought, okay, let's go down to 10 to the 3. And so at the end of the experiment, and that was 120 animals. And so we couldn't hold all of those animals in containment. And so we moved to, we only retained those 10 to the 3 animals. And so what we did was we wanted to look at the long-term antibody dynamics. And so these are the same animals I just described, but it's the 10 to the 3 group. And so what we did was we just monitored their antibodies for a whole year. After a year, we re-challenged those animals to see if they could ward off the infection. And then after we did the secondary infection, we continued to monitor them. So pretty straightforward. And so these are the median results. And the black line just shows the antibody results. And so to get you oriented, the x-axis is just the signal to noise ratio. This is a simple ELISA test, and I have it inverted, so that the low, so the beginning is no antibodies, and then it rises really quickly after they are infected. And so on the y-axis, it's just the day post-inoculation going out for the full year of testing. And so we see a very quick rise. On average, we look at this a lot. On average, the animals show an antibody response around day 7, often peaks around 10 days. And it's pretty tight across many experiments we did with it, detect anywhere from 5 to 7 days, usually by 10, and then it declines really quickly. In the natural exposure of wild animals, they're exposed to multiple viruses every season. And so you would hardly ever go a year without getting a secondary exposure, but that's what our experiment is showing. And so anyway, so we get this very quick rise, but then we get a very quick, rapid decline in the detectable antibody levels. I have these two levels here because we use this commercial kit. It's designed for chickens. Almost everything is designed for chickens or poultry. And so with poultry, they're looking at herds or flocks, whereas in wild animals, we're looking at individuals. And so we actually did a separate analysis to kind of optimize what the threshold for considering a positive would be. So the commercial assay is that solid line, and the dotted line is what we consider to be the alternative or optimized threshold for wild animals. And so if you were using, and this is a very commonly used test, but you can see that in either case, if we use the recommended threshold, those birds would appear negative. Half the birds would appear, the median result is negative after only 56 days. But even by the alternative threshold, the median response is that they're negative, by 112 days. We see this little bit of rise, it goes down, and we think this is due to the timing of our experiment where we inoculated in the fall to try to mimic dynamics in the wild when they're mostly exposed in the early fall. And so we think this rise is being driven by the females because they do a little bit more investing of resources to get ready for breeding and so that they have maternal antibodies. That's just our hypothesis. This is the exact same information. I just wanted to show the individual heterogeneity when you see the individual ducks. And what we see here is some individuals, if you can kind of pick out the red squares, you can see that that individual had a high antibody response across the whole year and was one of the rare individuals that was positive at all time points. And then there's other animals that were like, it dropped very low very quickly and they were negative almost immediately and stayed negative. And then some animals kind of popped up and down across the year of sampling. And so after that year, we reinfected them. And I don't really mean for you to read any of these individual numbers, but we inoculated them. And basically the result was, even though the animals were negative for detectable antibodies, a year later they had complete protection. We did not see anything. We had a couple of little blips, but that's normal just because we just put the virus in their throats and then we can detect it as it comes out and it's their way. So on day one, you get a little bit of blips. But anyway, so none of those animals became infected. So they were still fully protected a year later, even though we couldn't detect those antibodies. So clearly they had memory cells and they could ramp up. And so this chart shows the original data and then post the reinfection. You can see the antibody response after that reinfection, which is indicated by the vertical dotted line. And so that's the secondary exposure. And what we see is a true amnestic response whereby the animals have a very rapid boost or rise up of their antibody levels. So they were mostly negative, but then within 2 days, we saw that they became highly positive again. And then, so typical of an amnestic response, not only was it very fast rise, but it's boosted, so it's at a higher level than the initial exposure. And then we do see a decline again, but that decline is much higher than it was after that first response. And then interestingly, we see after that 6-month period, we again see that we see a rise associated with the breeding season. So just to break those data out, this chart just shows the same thing, but I've separated males and females. The females are in green and the males are in blue. And so what you can see is that those females always have a higher mean compared to the males, except for at the very initial, right after the exposures, but then they separate out pretty broadly. So I think that's pretty straightforward. Our antibody levels peaked at 14 days, and then we see that rapid decline. We do see strong individual heterogeneity across individuals. The females have higher levels on average than males. What we were seeing there was a combination of the inoculated and the contact ducks, and they had very similar antibody responses. Like there was no difference. I didn't break that out in the charts, but there was no difference between those individuals. And the clearest amnestic response after the rechallenge, where they had complete immunity a year later, even though we didn't have those detectable antibodies. And so because of this, the inference we can make is when we see this very strong immunity to that same virus, that we can assume that the only way this virus can persist on the landscape is if we get new recruits into the population. So the next thing I'm going to talk about was another subsequent experiment. And what we were really interested was transmission. And we're interested in heterogeneity across the infection curve. You know, a lot of times when we model transmission, we assume a very flat line and the probability of transmission, you know, our beta. And so we wanted to look at how does that actually change across the infection. You know, we can kind of assume that when we have a low viral dose, probably it's low and then it's high, but we wanted to actually get some quantitative data to evaluate that. And so our hypothesis is that, well, okay, and the other thing we want to get from that is we want to look at environmental transmission, because when we did this experiment, there hadn't been a lot of experimental evidence. We assume that the wild birds are getting it from water, but there hasn't been a lot of studies to look at that. So we assume that we have environmental transmission and that it's the primary driver, but we also want to use this transmission experiment to compare direct contact versus water contact. And we think that environmental transmission, because it's a fecal oral disease and these are dabbling ducks, that's probably how this happens. And so we wanted to estimate the time-specific probability transmission, and then we wanted to compare different sample types for, you know, what, like for designing future experiments, what sample types are going to give us the best information for looking at transmission, so we don't always have to test 4 swabs per time point. And then we wanted to look at the importance of time-varying transmission for SIR dynamics, which I'm not going to present here. That was the modeling that we did. I'm not going to present. I'm just going to look at the experimental results. The other thing that we were able to do with this was to estimate the minimum time until an infected contact started shedding. And then also the minimum infectious dose, which I'll get to. So as far as our methods, this is based on the experiment that you saw, and we selected 10 to the 4 as the dose we wanted to use for this experiment. And not because we thought that that was the best dose, but because it was the most reasonable with respect to sample sizes. That 10 to the 3, the dynamics were so slow, we would have had to use like hundreds of ducks, and 10 to the 5 happened so quickly, it would take more animals, because when they started shedding at 10 hours, I would have had to change out the animals every 10 hours to do the experiment. And so 10 to the 4 was kind of the in-between dose. And so that's how that got selected. And so for each mallard, what we did was we inoculated a mallard, and then we added 4 naive contacts. And then we added those contacts, and then at regular intervals, we removed the contacts, put them in individual housing to see if they became infected. And then we added 4 naive contacts. And then we removed them, and then we added, and then we removed. And we replicated this experiment for 5 focal ducts. So we did it 5 times. And so here's a schematic of what that looked like. At time 0, we inoculated the first focal duct, and we added those 4 contacts. And then 17 hours later, and this is what we determined for that from that 10 to the 4 data with the fine scale data, that that the first replication, the first excretion of virus was at 19 hours. And so we removed the ducts at the contacts at 17 hours to make sure that if a duct became infected, it became infected from that focal duct. And so we did this regularly. And so then we added 4 new, took those 4 out, added 4 new, took those 4 out. For the first 4 groups of ducts up at hour 74, we did that at every 17 hours. We used a 17-hour period. But after that, we had to space them out a little bit more. Just again, just the practicality of not using a gazillion ducts. And so after those first kind of 5 days, the intervals stretched out a little bit. But the basic idea is that we put some animals in, took some out, put them in, and then we could look at whether or not those contacts became infected. and whether or not the probability of becoming infected changed over the course of that infection. So here are some of the basic results. On the upper left, we see the number of birds. Remember, there's 4 animals for each group. And so between 0 and 4 animals could become infected for a different contact group. And then on the x-axis, I have the contact group. And so that's basically an indicator of time. That first contact where they were there for the first 17 hours. The second group was there for the next 17, well, actually the 19, because we had a 2-hour transition. And so what you could see is in the first group or two, not all the birds became infected, but then once we got to the third group, they all became infected. And then once we got later in the infection, the probability of transmission started going down again, and then none of the animals became infected. Well, we had the one errant duck who became infected. And then in the lower right, what we're looking at is the contact group on the x-axis, and basically that's looking at time. And then we had looked at what the concentration in the water was. So that basically what this is looking at is the probability of becoming infected at different concentrations of water. And so once we got to, kind of 10 to the 2, 10 to the 3, we almost always got transmission. And then once the excretion from the focal duct, and via that excretion, the contamination of the shared water, then we saw that the probability of transmission started to go down again later in the infection. And so I think I should have mentioned that. So we took oral, cloacal, and fecal swabs, but the animals had shared water. And so if the focal duct was using that water and excreting feces in the water, then the water became contaminated. And so another complexity here is that there is individual variation in behavior. And so in some of these groups, the focal duct spent quite a bit of time in the water, and almost all the feces went into water. And then we had one individual that didn't use the water. And so that adds to the heterogeneity that we see across this experiment. And so I'm going to kind of skip over, but we did model that probability of infection across the time period, and we also evaluated what was driving the probability. Was it the exposure, the dose in the water? Was it the dose in the oral swab, the dose in the cloacal swab, or the dose in the fecal swab? And so we're using the oral, cloacal, and fecal as indicators of direct contact, and then the water as indirect contact or environmental transmission. And just with some basic modeling, what we see is that the concentration in the water is the primary driver of the probability of becoming infected. And so this tells us that this is a support for our hypothesis that in nature, environmental transmission is the driver, not direct contact. We were also able to look at the minimum infectious dose, and what we found was that minimum infectious dose was just about 10 to the 2. So that's pretty low. So animals exposed to a very low dose of virus in water will become infected. And so I think I went through that pretty quickly. So this is kind of the same thing. Water is the primary driver. The transmission varies over time. Mallards can become infected at a pretty low dose of virus, and that kind of goes to this whole thing, like they are so well adapted to this virus, and you see those quick kinetics, low exposure dose. And again, everything we do with these dynamics of this virus is that there's always lots and lots and lots of heterogeneity. And so it's hard to just look at 4 or 5 animals when we do these experiments. We really need, 10, 20, 30, 40 animals to really get an idea of what that variability looks like in a good mean estimation. We needed to go there. Oh, I guess I thought I took our modeling out. There's a lot of heterogeneity, but I'll leave that. A lot of heterogeneity, and it did matter. It does matter if you use a flat transmission versus accounting for time across that infection. And so I don't want to have plenty of time for questions, so I'm going to end right there.
That's great. Thank you so much. It's wonderful to see high frequency sampling of viral loads. People in this room are always complaining that people don't ever sample viral loads on a frequency that allows you to do decent modeling and parameter identification. And so to see you doing 2-hour samples is already inspirational.
Yeah, it's hard to find people who want to spend the night with me in those duck pens.
I hear you, but it's really nice to see. Okay, well, time for some questions. We have 20 minutes for questions. Veronica.
Oh, so I have a few questions. So for the first part of the talk, so you show the data, and this is very interesting and very puzzling. You show that inoculated birds and transmitted birds, they have almost like identical curves for viral load, right? And at the same time, you showed that, so the first question, did you actually shift, or it's in real time exactly the same, or did you shift the transmitted birds slightly, like, or to
I see your question. Let me see what I'm presenting here before I answer that.
yeah, it's
I just to see which, I've done this talk a couple of times. Yeah, I think this is the actual, I'm showing the hour post inoculation, because sometimes when I show it independently, I'll show that hour post exposure. But I think it's, if you look in that first, going back to this first graph, the 10 to the 3, you can see the slight offset. It's hard to see it at this level because I had such fine scale, sampling, and you're not seeing it. So they're only, like for that 10 to the 5, they're only 10 hours apart.
so it's basically real time after inoculation for both, for inoculated and for transmitted. And then you show on one of the next slide that actually shedding was delayed for transmitted birds. So I'm just trying to, how could we explain that the viral load is exactly the same, but shedding is so much delayed for transmitted? Because
well, I think the thing is, so if you look at this one, it looks like it's almost exactly the same, but keep in mind, we're looking at 10 days. And so that very small shift you see, the gray birds, the gray line is the inoculated, and the orange is the transmitted. And so it looks like it's almost on top, but that is actually that kind of 10 hours later, right? The 10 to 20 hours later. It's just that we can't see it because I'm showing 10 days. And so it's very tight. And that's actually why I showed this, because you can see it much more stretched out when you see the fine scale data. So I think the shift is there, like that the transmitted birds are just a little bit later. It's just that I'm showing all 10 days of the experiment. And so it's just very narrow in this picture. Does that make sense?
Yeah, I can see it now. It's just,
yeah, they're very similar, but I think that goes to just how quickly these dynamics are happening, right? That we are seeing in less than 24 hours, we're seeing the contact bird begin to shed. It's pretty incredible, I think.
and did you try to estimate the half-life for antibodies for the decay? For the decay after first, for the decay after second, because it looks like the slope is different after second. So
yes, I would agree with that. No, so one thing I'm going to say about this experiment is I think these are really, I'm very interested in these experiments and these very fine scale data. But the day before I started these experiments, these were very intensive experiments. I mean, this is a ton of animals, a ton of sampling, a ton of, like, these are more than 5,000 samples we had to do in the laboratory. We reorganized and I lost all my staff. And so I'm trained as a quantitative ecologist, and I have just never gotten back to these data. So I have these experimental data. It took me 6 years to get the laboratory testing done because I had to do it with work study students. There's a little bit of better background. And so I'm just started working with a modeler actually just like 2 months ago to try to not let these data just sit in a file. But I had, so the intent was that we would do, I wanted these parameters so that I could model them. And so I had to do the experiment, but kind of my focus and my reorganization, I have different priorities. And so we're just starting to get back to these data to do the modeling. So no, I haven't done it,
Yeah,
but I would like to.
very beautiful data. It's like a dream for people who is doing this type of work.
Yeah, I think everyone who does experimental work should be trained as a modeler, because you design your experiments very differently, I think, than other people.
Yeah, like our group, we analyzed the decay rates after influenza infection and after COVID right now, but mostly in humans. So it would be kind of very interesting to compare it.
Mm-hmm. Yeah. So actually, I don't know if I have it here, but we also, I was a little bit concerned when I saw those antibody dynamics. Oh, I don't have it here. I was a little bit concerned when I saw those antibody dynamics because I was using that 10 to the 3 groups, and I didn't know if those antibody curves would look different based on the exposure. And so I was immediately traumatized. I was like, oh, maybe I should, but the animals were euthanized. There was no going back. And so I did actually do a subsequent experiment where we looked at 10 to the 3, 10 to the 4, and 10 to the 5. And then I looked at those antibody dynamics. And it was more designed around the antibody dynamics where we did 10 to the 3, 10 to the 4, and 10 to the 5 to look at whether those antibody kinetics looks the same for different exposure doses. And then we also specifically looked at males compared to females. And we specifically looked at old versus young, because we have, what we see is, I don't know if you're familiar in ecology, there's this investment trade-off. And so the young animals actually have a stronger immune system before they become reproductively mature. And so we wanted to compare, so we used 6-week-old animals and 6-month-old. Ducks are reproductively mature at 6 months. And so we did kind of a designed experiment with those 3 results. And what we saw from that is that the antibody dynamics did not vary with that exposure dose, just in case you're curious.
That is very interesting, because we might expect, at least from the humans data, that, for example, if you compare 10 to the 3 and 10 to the 5, then for 10 to the 5, you probably have more antibodies, like at peak, right? Or you have the same. I don't know.
Yeah, I'm gonna see if I happen to have that.
The initial decay will be faster. That's what we would expect.
Yeah, we only went out 6 months in that experiment, but my memory is those curves were almost, I don't have it. I thought maybe I could find it. I don't have it open. For the doses, we didn't see any significant differences. And I just did simple statistical tests comparing those curves, but they were the same.
So even they generated similar amount of antibodies at peak.
I'm sorry, you kind of faded out.
At peak, there were like the same amount of antibodies for 10 to the 3 and 10 to the 5.
Yeah, I'd have to look at it. We haven't done the more extensive modeling because this is kind of where we reorganized. And so I didn't do more extensive, but like when you plot them, they look almost exactly the same.
Thank you.
Sure.
I mean, that's fascinating that the re-exposure seems to have slower kinetics. I think Veronica was seeing something similar in the data set she was analyzing for humans for post-vaccination for COVID, where
these experiments are so much more interesting. This kind of thing, when I was doing it, like no one really cared, but because we would love to have these data for humans, but that's a little harder experiment. Those are a lot more. I think I have to get a lot of permits, but obviously it's more difficult with humans. So anyway, we're starting with the transmission experiment just because we were furthest along with that modeling, but we're hoping to get to those the antibody data pretty quickly too. So I could share it with you once we get a little closer, or even I could probably pull that exposure dose up. That was actually just an undergraduate project, so she hasn't up yet. I have high demands of my undergrads.
you've got so many time scales in your experiments, right? You have these very fast time scales for eclipse phase, and shedding. You have the longer time scales for the appearance of the antibodies and then their decay. That intriguing recovery of the antibodies, which, Veronica, do you ever see that in humans, or is that something that's unique to ducks?
That's kind of unique. That's also very puzzling. And do you have any kind of mechanisms? Do you have any ideas about mechanisms?
Well, I think the difference is that as a species, we're reproductive all year. And so I think like in wildlife, we see, this is not with antibiotic dynamics, but there's so many things that we see that are seasonal, right? Because they only breed once a season. I mean, under optimal circumstances, ducks will rebreed, but they're mostly getting up for the breeding season. And they actually, so for ducks, they actually retract because they need to be light to migrate. They actually retract their reproductive organs in the fall. And so I think that that's what they're becoming. So even though they're reproductively mature at 6 months, seasonally, they retract their, that's why their plumage changes seasonally for breeding birds. And so they retract their reproductive organs and then they invest in those reproductive organs. And so it could be, so there's that idea. It could be that the females are investing for those maternal antibodies. So another experiment we did was to look at those maternal antibodies and how long we could detect maternal antibodies in chicks. And it's pretty short, actually. It's pretty brief. It's only a couple weeks. And it really peaks around 10 to 12 days, I think. Don't quote me on that. I have to look at the actual data. But anyway, so I think that is just a function of them being, annual breeders as opposed to regular breeders like we are.
I had one short timescale question, which you alluded to, but then I didn't see the answer to, which was how long you have active virus after it's shed in water. How long does the water remain infectious? Because RNA by itself doesn't tell you how many infectious viral particles you've got. And so
yes.
do you have any sense of how, after the water is contaminated, how long it takes, or how long the water remains infectious?
So people are starting to do that work and do it more intensively. But I think it's longer than we think. And I just want to put on my virologist hat, I'm not a virologist, but I'll put on a virologist hat. And one thing that we see specific to influenza is that, with this extremely high mutation rate, what happens is a very high number of those particles aren't infectious. We've been focusing on infectious particles, but what more recently, if you go to virology meetings, more recently what we find is that because we get a population of particles, that I can have two non-infectious particles, but if they co-infect, they can infect, right? So we've had this strong focus in influenza research where people want it to be infectious, but really they have to look at that whole population of viruses because you could have a fully non-infectious group of particles, but they're still infectious as long as you take in enough of them and they complement each other. So I just kind of want to make that point as a side note.
Right. So as long as you get one of each of your 8 RNAs, you're infected.
Yeah, as long as you get all those genes represented in the same cell.
Somebody 2 years ago at the beginning of all this was telling me about, I think it was a cow disease, a cow virus, where actually there were 3 separate virions. So the genome was actually normally packaged in 3 separate virions. You had to be infected by all 3 independently to get the disease.
Yeah, so I think that's relatively new. And so it kind of, just because a lot of people will pick on when you're using PCR about RNA, but when you're only looking at infectious virus, well, then you're missing possible infections. So, but a lot of studies show, but most of those experiments have been done and they're not using natural bodies of water where there's all kinds of things that might impact that. But that show and it varies by the strain, it varies by the temperature, it varies by everything. But often it can be for days. The water can be infectious for days. And even the virus, like there's a recent paper, maybe last year, where he found infectious water samples and then it freezes, and this was in Alaska, and it was still infectious a year later. I mean, that was probably because the water was frozen, but that it was infectious a year later is pretty impressive. Excuse me.
Other questions? I think it's. I certainly was. I was taking a lot of notes. I'm trying to see what. There were a couple of other. I had a dumb question, which was in the signal to noise. Was that a log scale at the y-axis, or was it or a linear scale for the decay?
I think it's a linear scale. My memory, I'm so involved. The last 2 years has been all SARS-CoV-2. So, but going back to that ELISA that we use, it's just the absorbance that comes off the plate reader, and then you're just subtracting out the difference between the positive and the negative. So I'm pretty sure that's a linear scale.
Okay. Time for one last question, two last questions.
And whether this data published or not yet? This key data, were they published already?
No, that's what I was saying. That none of this is published. It's been languishing. It's languishing after my reorganization. And we just started working on the modeling piece. You know, hopefully we'll get it out. And then our agency, we're emergency response. And so when you have SARS-CoV-2 in wildlife and you have high-path AI outbreaks, I get taken away from my position to do emergency response. And so I would like to get it out, but maybe this is, I saw in the original email, like maybe we need collaborators to help us get some of this out. But it was kind of languishing for a while, and we just picked it up a few months ago. So, I mean, like, I think I gave these talks like around 2014. So that's the impact of a reorganization.
Well, it's beautiful data sets. It's really very inspirational to see these data. And your comment that experimentalists should be trained in modeling was very, very appreciated by this audience.
Yeah, and you know, well, and I think it goes both ways, because I'm kind of I'm so much at the intersection, and you know, and I just see so often that, you know, people who are they just don't understand the modeling, like just talk past each other so frequently, you know, and like just really trying to communicate the utility, you know, like why would you model? Like I've had someone say, why would you model the data? You have you have the real data. It's like, well, the real data, you know, can be biased. So it's kind of interesting to work at the intersection for me. And really, I was trained as a modeler, but I've been doing these experiments because there were no data for me to model. And so I had to just learn how to do the experiments.
Okay, any other questions for our speaker before we? I hope you'll come back another time and maybe tell us about some of your other projects too, or just show us the results when you got the modeling.
These ones, yeah. I mean, we're hoping to get this one. We have pulled in like a postdoc who's now helping us try to get that transmission one out. But yeah, I don't want to just publish these as raw data, because I feel like there's a lot more we can do, and hopefully, if we don't keep finding SARS-CoV-2 in wildlife, I can get back to my favorite pathogen.
Ruchira, question.
Yeah, you said you have undergraduates. So how is it training them? You're doing both modeling and the experiment in your lab. So do the undergraduates do one or the other, or do they cross over? How do you go?
Just because of
a project that they do for like one internship or something.
Yeah, usually I've either worked with an undergrad that's helping with modeling. We actually, so Fort Collins, we have Colorado State this year, and a couple of the ways we've done that is they have like a lot of universities have that. The stats department has a consulting class, And so they'll work with our data set as a class, so I'll contribute one of these data sets and they work with it. So on the modeling side, that's mostly what I've done. I haven't had a quantitative student. And then, and so usually my students, I'm in a wildlife group, so they just want to handle animals. They don't want to do math. That's the quick answer.
Okay, thanks.
Well, Colorado State in the physics department has some very good modelers. So they might actually teach, I'm surprised they don't teach a course in network dynamic models. Which
they have
could be
a lot
ideal
of theoretical ecologists as well. It's just that the way I get students isn't usually through those departments. I usually get my students through the wildlife department. And I don't have a tremendous amount of students. It's harder federally, because I'm usually using appropriated funds. I don't have guaranteed funding for year-to-year funding. And we're very applied research at our center. And so it's harder to get a grant because I might have ecological questions or, more academic questions, but they want me to do my mission research. And so it's a little harder for us to have. So we usually have like an undergrad or a postdoc, and there's not a lot in between.
No, but when you said maybe have a class project, a semester class project for class to do the analysis, I was just thinking, I turned my ODE class in networks to doing SIR type and modeling for the undergrads. And I was thinking, boy, that data set would be a wonderful class project. How do we do that? How do we do SIR models in-host versions of SIR models? How do we model antibody production and release and decay? And then how do we actually fit real data? It would be sort of an ideal semester-long class project for a group of motivated undergrads. And I think Colorado State, they'd have people who would like to do a project like that. But yeah. Maybe we can, when I teach it in the fall, if you still haven't done it all, maybe we can talk about that.
Yeah, feel free. Right now, it looks like I'm going to be doing a lot of emergency response. So feel free to contact me. We were just starting to get it going when the high path came. I thought I was kind of getting SARS behind me, and then the high path has been a problem. Not a problem, but a distraction from getting to ever model my data.
Joy, question.
Not really a question, just a comment. That was a Herculean effort you just showed. I'm a cellular immunologist, and holy cow, that was amazing. But I wanted to laugh at your comment about the ducks that won't behave like all the rest of the ducks, because I work with mice with influenza, and we're using a voluntary running assay to measure just voluntary activity, and 99% of the mice will happily run on the wheel all night long. And there's one dang mouse who will build a nest on the running wheel. So I felt your pain in that experiment.
Yeah. I mean, and for me, that's just always been anecdotal, but the more we've been doing these experiments, the more we realize that we should probably have a behavioral, component, because sometimes that's going to explain that heterogeneity. It's just they have very behavior, they're just very different.
Temperament.
Yeah.
Darn it, they will not just all act the same for us.
No, certainly not.
Okay, any other questions or comments? Okay, well, I want to thank Susan again. That was a tremendous inspirational talk. The fact that you started out doing modeling and you had to learn all of that and do those incredibly hard experiments to get the data, I mean, it's a tribute to you, but also a statement about how much missing data there are. Also, it was really exciting to hear that. Thank you so much for sharing it with us, and I hope you'll come back again and tell us about your progress and other projects.
Okay, well, I appreciate it. It was great. Thanks so much. I really appreciated the questions.